Hello, everyone, and welcome to today's live broadcast, Single-Cell Sequencing to Investigate Epigenetic Mechanisms in Inflammatory Lymphocytes. I am Jen Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Illumina and Dolomite Bio. For more information about our sponsor, please visit their websites at illumina.com and dolomitebio.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenters, Dr. Thomas Ayers, Dr. Pawel Zajac, and Dr. Adam Cribbs. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Dr. Ayers, you may now begin your presentation.
So hello everyone, my name's Thomas Ayers. I'm a technical application specialist for Dolomite Bio. Dolomite Bio is a company which is dedicated to providing microfluidic solutions for single-cell biology. And today I'll be taking you through just some sort of groundwork so that we're all on the same level when it comes to knowing about single-cell RNA sequencing, so that when we get into the real meat of Dr. Adam Cribbs' research, we all know what we're talking about. So first of all, I'd like to go through single-cell RNA sequencing from the perspective of performing it with microfluidic devices so that we can do so in very, very high throughput. This abbreviates to scRNA-seq. So what I'll be talking about here is how the work for single-cell RNA-seq, some of that has been performed on the Nadia platform within the paper that Adam Cribbs is about to present. So I'll go through the workflow now of single-cell RNA-seq on this Nadia platform using microfluidics. First of all, we start, as with most single-cell workflows, by making sure that we have cells with very, very high viability and cells which are adequately dissociated from each other so that they can be separated into single compartments. From here, we move on to the aspect which is performed on the microfluidic device, aka the Nadia or the Nadia Innovate. And what we do there, using this microfluidic junction in the center, is that we are flowing cells within cell buffer and oligo-DT capture beads within cell lysis buffer together into a microfluidic junction, which is then cut off by a very high-speed oil jet into individual droplets. This happens at around anywhere between 1,000 and 2,000 droplets per second, and so we can get very, very high throughput from this. The result of this is that we end up with droplets containing just one cell and just one of those beads. And on those beads, we have many, many oligos which contain a barcode sequence, the same for every bead. The same for every oligo on one bead, so that we can later identify which read is coming from which cell and bead. And also we have on each of those oligos a poly-DT capture tail so that we can capture the polyadenylated mRNA. The next step of this process, which also occurs on the Nadia platform, is that we undergo cell lysis within those droplets. So we're breaking the cell open, breaking out the mRNA, and the mRNA then binds to the poly-T tails of the bead. At this point, with scRNA-seq on Nadia, we do something very special, which is when we break open the droplets and take all of those beads into basically a bulk situation in a tube, so that we have all of those beads with all of the mRNA captured on it, all sitting together, at which point we can go on to the next steps of library preparation. Library preparation involves basically doing reverse transcription to generate cDNA and then amplifying a library off of that using PCR. Then we can use different tagmentation kits such as the Nextera kit provided by Illumina to make these libraries compatible with next generation sequencing devices, which Pawel will go more into later. After which we have all of the read information necessary to undergo all of our complicated bioinformatics and take all of that information, decide which cell, which read came from, which transcripts are produced the most, and we can create some very interesting diagrams to do with what kind of cell types come out of your particular tissue of interest. So that would be the workflow of general scRNA-seq on Nadia. But what we can go through just quickly now is the Nadia product family and how it is unique in terms of a microfluidic droplet-based fluidic device. There are a few different unique aspects to this, the first of which is our proprietary technology here, which we have proprietary stirrer drives within the chip itself. This is where we have magnetic stirrers within the cell and bead reservoirs, ensuring that all of our cells and beads are fully suspended in solution for the entire run. You can imagine if we've got the cells coming through in clumps where they've sunk during that microfluidic process, that can result in doublets. So the stirrer drives on the Nadia are there to prevent that. We also work with 3 independent pressure pumps for cells, beads, and oil, so that we can do some quite interesting things later on to do with changing pressure levels to account for different buffers and things like that if your cells are happy in different buffers. And finally, throughout this entire process on the Nadia uniquely, for a microfluidic droplet-based device, we have temperature control using a Peltier drive, allowing us to keep the cells and mRNA at 4 degrees, avoid mRNA degradation, maintain transcriptome state throughout that entire process. This temperature control can also go up to 40 degrees, but that's really becomes interesting with other different kinds of applications, which you can read about on our website. So overall, we know that with the Nadia instrument, we can use those standardized protocols for scRNA-seq in order to generate high throughput single-cell transcriptomes using that application and using that workflow. We can do so with scalability up to 8 samples at a time, and we have various other benefits coming from the fact that we're using those microfluidic chips, which are disposable themselves. Just very briefly, what I'll do is I'll move on to the added module for the Nadia Innovate. So this is an add-on which sits beside that Nadia machine, which I've just shown you. And effectively what that allows us to do is to port the pressure from those 3 precise pumps from the Nadia into a single chip. And we can view the dropletization process of creating droplets and flowing cells and beads into them in real time using the high-speed digital microscope camera. What this allows us to do with the integrated flow control software is that we can play with different settings to do with pressure, to do with temperature, and to do with stirrers to see whether we can maybe improve on the microfluidic aspect. So if you've got, let's say, big cells, we can decrease oil rate, things like that, in order to get the droplets bigger. If you've got cells which are only happy in a particularly sticky buffer, something which happens a lot when you get towards niche cell types, then we can account for that buffer thickness by altering the pressure levels of those different buffers. What this allows us to do, basically, is to open up the entire system to optimization of existing protocols. So we can take that scRNA-seq protocol that I've just talked about, tweak it to make it best for your particular cell type, or we can even create entirely new protocols or single-cellify existing bulk protocols and things like that. We can do so in real time using that high-speed digital microscope and camera, and that we can then port these protocols back onto the Nadia system to run in an automated way with up to 8 samples at a time. So without further ado, I will now move on to and allow Pawel to take over, where he'll take over the part downstream from encapsulating those droplets and through to the aspect that takes place on sequencers once we have a sequenceable library. So over to you, Pawel.
Thank you very much, Tom, for this great introduction to the Dolomite biotechnology and to the single-cell sequencing workflow. So, as mentioned, my name is Pawel Zajac. I'm part of Illumina's marketing team, and it's my great pleasure to, within the next 5 minutes, present who we are, what we do, and a couple of our recent news. So, for those of you who haven't heard about Illumina, we are a global company, and we develop, manufacture, and market innovative next-generation sequencing and microarray technologies. And our technology enables you to make foundational discoveries to transform medicine, and ultimately to impact lives. And to date, there are more than 200,000 publications where our technology has been used. So it clearly shows that this is a robust technology that you can trust. And there are two ways in which you can get access to this technology. You can either purchase a system, implement it in-house in your own lab, or you can turn to one of the many service providers that are available and get access to our technology there. And as mentioned, one of the technologies we offer is next-generation sequencing. And it is a single technology, but it unlocks a wide range of applications, a wide range of biological insights. So the sequencer can be seen as a tool, and depending on how you prepare the sample, you can use it to study many different facets of biology. So if you take, for example, a look at cancer research, you can, of course, use sequencing to perform single-cell analysis, as is the single-cell analysis topic of this particular webinar. But you can also do tumor mutation profiling, figure out what differentiates the tumor from normal tissue. You can study the microbial component to study the interplay between this component and tumors. So a single technology, but unlocking a wide range of applications. And we provide sequencing systems that enable a wide range of single-cell applications. So no matter which biological modality you're interested in, no matter if you're interested in doing a global study, for example, studying the entire transcriptome, or if you want to zoom in on a particular subset of transcripts, we do have a sequencing solution that fits your output needs. And it's exactly the same with study size. So from small studies to very large studies, we are more than happy to support you with the sequencing part. And here we also show our portfolio of sequencing platforms. So on the left-hand side, you see the iSeq 100. That's our smallest, most affordable benchtop system. And with 4 million reads, it's too limited for the majority of single-cell applications, but it's a really good fit for quality control of libraries before you run them on one of the larger systems. On the right-hand side, we have the NovaSeq 6000. That's our production scale sequencer for those large studies. And what I wanted to introduce now is our latest sequencer, the NextSeq 2000, which allows deep, large studies, but still within a benchtop platform. So there is a lot of innovation that has gone into the NextSeq 2000. So it clearly shows our long-standing tradition of technological advances. And all these innovations translate into significant gains. For example, there is a 2x reduction in operating costs compared to the NextSeq 550 instrument, and the output has been increased from 400 million that you get with 400 million reads that you get with the NextSeq 550 to 1 billion reads on the NextSeq 2000. So opening up for those larger and deeper and wider studies. And we have tested the NextSeq 2000 on a wide variety of different library types, and it gives the same high quality data that you've come to expect from Illumina. So for example, if you take a look at the figure to the right, you see that there is a really strong correlation between the NextSeq 550 and NextSeq 2000 for mRNA-seq libraries. So the same high quality data that you've come to expect from Illumina. Our production scale sequencer, the NovaSeq 6000, it has also seen continuous innovation since it was launched in 2017. And a couple of months ago, we released the latest version of the reagents for this instrument, and they are called version 1.5. And these carry a number of gains. So one of them is an reduced price, which means that you can sequence more with the same budget. So this once again opens the door for those larger, deeper, broader studies. For example, if you want to add a modality to your existing single-cell experiments. And in addition, the consumables also have a greater flexibility, improved robustness, and enhanced user experience. And one example is that the shelf life has been increased from 3 months to 6 months. And with this, I wanted to thank you for your attention, and I encourage you to get in touch with us. So if you are planning a single-cell sequencing experiment, don't hesitate to get in touch so that we can have a discussion and propose to you the best sequencing solution for that particular project. So with this, I wanted to once again thank you for your attention, and I will now turn the presentation to Dr. Adam Cribbs, the star of the show. Please take it away, Adam.
Thank you, Pawel. Morning, afternoon, and evening. I'm Adam. I'm group leader in systems biology at the Botany Research Centre within the University of Oxford. And I lead a computational biology team with a broad interest in systems biology, immunology, and epigenetics. Today, I would like to give you an overview on how we've been using single-cell transcriptomics to understand more about the role of Jumanji histone demethylases within the immune system. So the first thing to mention is that I have no disclosures. Unfortunately for me, but fortunately for you, I'm not being paid to deliver this talk. Therefore, I can talk very openly about my own research, but also I'm happy to provide questions on the application of the technology we've been using to address some of these scientific questions. So just as a bit of a quick background to myself, so my background is actually as a molecular immunologist. I undertook my Ph.D. At the Kennedy Institute of Rheumatology, but during my postdoc, I actually started to generate large amounts of sequencing data, and I realized that I had none of the skills to analyze any of the data. And so I ultimately relied on other people within the group, and not having ownership over my own science or the ability to analyze the data was actually the most frustrating time I've ever experienced in my scientific career. And this ultimately acted as a catalyst for me to successfully apply for a fellowship to completely retrain in computational biology with Professor Chris Ponting, also within the University of Oxford. And it was actually here that I developed advanced skills in a number of statistics, software development, and data analysis that have allowed me to undertake large-scale data-driven projects. So now I'm actually leading my own computational research group, and the focus has definitively shifted towards single-cell within the past 2 years or so. So colleagues and myself have been involved in developing novel single-cell methodologies, both wet lab and on the computational side, and unfortunately I won't have time to go into that today. But I also lead the computational component of the Tendon Seed Network, which is part of the Human Cell Atlas project, and coordinate the computational analysis teams involved in applying single-cell sequencing to multiple myeloma clinical trials. More recently, I've been involved in working in a large team of scientists, playing a small role, but an important role within the single-cell immune profiling of COVID samples as part of a larger combat consortium within the University of Oxford. However, what I'd like to speak to you today about is how we're actually using single-cell sequencing as a tool to investigate epigenetic research. Specifically in this talk, I want to give you an overview of the single-cell technologies that we work with, an overview of maybe some of the quickly over the positives and negatives of each. And then I'll introduce our computational strategy we take to analyze the data. Then, using an example of the Jumanji histone demethylase in recent published work, I'm going to show you how we can use single-cell technologies applied as a research tool within to answer scientific research questions. Initially, I'm going to give an overview of the single-cell sequencing state of play within our laboratory. We have essentially 3 systems, which we routinely run within our laboratory. The first is SmartSeq2, which is a well-based approach where single cells are fact sorted into individual wells in plates, and then chemistry takes place within those individual wells. The 2 other systems are droplet-based approaches. This is the Nadia system, which Tom introduced before, and the 10x Chromium system. So each technology is applied really to address different scientific questions, and each technology has its own different quirks and advantages and disadvantages over the other. So when you look at, well, actually, I suppose the question that I get asked routinely by members of the group is which technology should I use to answer my research question? And the answer isn't actually always that easy to simply address. However, when you compare droplet-based systems against SmartSeq2, or actually many other well-based approaches, the issue that you will always find is that there's a trade-off between cell number and the numbers of genes detected in your analysis. The cost of SmartSeq2 and the fact that can be a very manual process means that SmartSeq 2 cost per cell is very high, although it gives you superior resolution in terms of the gene expression. So it's quite high depth in terms of the numbers of genes. When comparing Nadia and 10x, the comparison is pretty much always due to cost versus cell numbers versus the ease of protocol. Firstly, the main thing is that the principles between DropSeq and 10x are very similar with slightly different executions. The Nadia system is around half as expensive as 10x, so it allows us to actually perform more sequencing experiments. However, the protocol is ever so slightly more hands-on for DropSeq. And another factor is that to generate good DropSeq experiments, you typically require more input cells. And I think in our lab at the moment, I would reckon around about 70 to 80% are actually ran on the Nadia system, and the rest on 10x. We are at the moment very rarely doing SmartSeq 2 because of the low throughput and the cost. So the single cell workflow is very simple when you compare it to the original DropSeq protocol. The encapsulation process is handled entirely on the Nadia machine. And I'm going to talk about the Nadia machine because this is actually what we've been using as part of the workflow for the T cells I'm going to talk about. There's no need to set pump speeds or any complicated pump setup like you would do with the original DropSeq method. The encapsulation is very simple. You have a stream of oil, a stream of beads decorated with oligonucleotides that capture RNA. This buffer also contains lysis solution, and separately you have a stream of cells. So once the cells encounter the lysis buffer in the bead, they lyse, release their RNA, and then are captured within the bead. The library is prepared as per the original DropSeq protocol. I think Dolomite Bio now have a modified simple protocol to increase yield, which actually we don't use in our lab, primarily because we have this working quite well, and I don't really want to change things when things are working. Then the sample is then sequenced, analyzed, and I'll go on to this in subsequent slides of how we analyze our data. So the design of the bead is important for this method to work, and it basically follows the original McCosco 2015 paper. You have a bead built up in the 5' to 3' direction, so you can capture the 3' RNA polyA site. Then all downstream chemistry is then performed on the bead, which includes a barcode that allows you to link the read back to a per cell and a unique molecular identifier. So then you can, which is specific to every individual RNA, and then computationally you can work out which RNA belongs to which cell and then remove PCR duplicates using the UMI. So I often see single-cell sequencing methods actually used on projects which don't need single-cell sequencing requirements. The first question that I always ask is, do I actually need to run a single-cell experiment? And will a bulk RNA-seq actually give me a better transcriptional resolution? And actually it'll be way more statistically robust than a single-cell experiment. And plus it's going to be a lot less expensive. Single-cell sequencing, however, is used best when it's used to address specific scientific questions related to heterogeneity, lineage tracing, or studying cells through a dynamic stochastic event. The most important factor, and something that I find quite often gets neglected, is that people leave the most important bit, the question of how you analyze their data, at the point when they've generated their data. This question really should be addressed first thing, because if you don't have the expertise within your lab to analyze this data, then you need to find solutions to overcome this. And if you get computational people involved early on, they can help inform how best to set up a single-cell experiment to avoid things like batch effects and get the most out of your sequencing experiment. A note of caution on using commercial computational approaches is that really you need some expertise, because these computational analysis tools will always give you an answer at the end of it, and it might not always be the right one. So DropSeq was actually initially analyzed using a set of DropSeq tools by the Macaro lab. These have actually been nicely written up into this DropSeq pipe SnakeMake pipeline. I've ran this pipeline and it does a really nice job. However, I've personally moved away from DropSeq tools because the field has moved on considerably since 2015. My preferred approach is something called pseudo-alignment. Instead of heavy computational mapping approach of DropSeq tools, you're actually aligning to a transcriptome, which makes the whole process less computationally expensive, fast, and more accurate than a traditional mapping approach. There are two implementations of pseudo-alignment. This is Callisto Bus Tools and Salmon Alevin. If you want to learn more about pseudo-alignment, I recommend you looking at the Bray et al. Method, who really lay this out quite nice and very beautifully. But which tool to use? So I've been benchmarking Callisto and Salmon, and what I find is there's a slight increase in the precision and recall with Bus Tools Callisto. However, in real life, there's actually no overall population change between the two methods. And I think the differences are quite negligible, really. So I really think that here, consistency is key. Don't change the way you process your data halfway through processing some of your samples. Keep it all the same. Personally, I choose Bus Tools at the moment. That was only purely because it was convenient at the time, and I saw a slight increase in the benchmarking in terms of the accuracy. Overall, our single-cell workflow is as follows. We use pseudo-alignment to align our reads to the transcriptome, and then use Bus Tools to generate counts of cells by genes. Quality control is a real big issue and is key here, and I use the SCADA-R package as well as bespoke scripts to quality check all of the data. I spend probably 70% of my time just quality checking data before actually doing any downstream analysis. All of these these statistics are then added to an SQLite relational database along with any metadata associated with the experiment. This database can handle millions of cells, and it actually scales really well. So the workflow can scale from thousands up to millions of cells that actually I'm working on quite regularly now. Then downstream analysis such as doublet identification, single R for classification, and visualization of t-SNE or UMaps are then performed and then visualized. My preferred method is cell by gene by the Chan-Zuckerberg Initiative. So we typically use ScanPy or Seurat for our downstream analysis, as well as bespoke scripts. And really the tool that I use largely depends on the complexity of the project and the numbers of cells. I'm slowly, for larger projects, Seurat and R-based methods just don't scale, and you need a ScanPy at the moment is what we're analyzing most of our data sets in, because this scales very nicely to millions of cells. So the lab workflows are, if you're interested, are all on Git, and the pseudo-alignment runs remarkably fast. And otherwise, I still would recommend DropSeq pipe if you don't want to use any pseudo-alignment approaches. That's equally, and in fact, actually, it's a really nice SnakeMake pipeline. So now we come to the science aspect, and specifically how I'm using single-cell biology to answer some scientific questions, specifically within epigenetics. The first thing that I'd like to emphasize is really how we use single-cell primarily. Single-cell sequencing is really just a tool that should be used within the wider scientific context of your other scientific tools. So the technology is actually really now matured in both the wet lab and the computational side, so much that we actually now run it very routinely almost every day. Therefore, single cell should be used within the wider context. And therefore, the first experiment I always ask myself, as I mentioned at the beginning of the talk, do I actually really need to run the single cell sequencing experiment? So the main considerations for running or not running a single cell sequencing experiment largely come down to how heterogeneous or how variable you think your tissue or cell population is. Are you working or are you wanting to study a dynamic process, and how variable are your treatment's conditions will ultimately improve, or you'll get a better chance of using single-cell sequencing technologies to understand this heterogeneity within your tissue. specifically looking at identifying composition changes or differential expression between the different subsets within your experiment. So our typical laboratory workflow in the group is we use chemical and biology with knockdown or knockout screening approaches to identify targets within cellular systems. I then leverage this with information and identification from molecular mechanisms underpinning these effects using a combination of the latest diagnostic techniques such as bulk RNA sequencing, single-cell transcriptomics, CyTOF single-cell proteomics, and very basic biochemical assays. What we're doing with these tools is trying to build a consensus to answer a specific mechanistic scientific question. We leverage all of this mechanistic insight with disease biology and have incredibly very effective clinical collaborators that make translating our basic scientific findings into disease-relevant biology very painless. So the project that I'm going to talk about today started from a small molecule epigenetic inhibitor screen that was performed on Th17 cells. We work very closely with the Structural Genomics Consortium within the University of Oxford, and their expertise lie within crystallography and developing small molecule epigenetic inhibitors to inhibit the epigenetic machinery. We then use these libraries of inhibitors within simple screens. So we now have a collection of libraries that target the epigenome, kinome, metabolome, ubiquitinome, and proteostasis pathways that are curated by Professor Uda Opperman, also within the Botner Research Center. These small molecule probe libraries are curated, and then they are constantly updated and expanded, and then used within assays. So we used our library of inhibitors. When we use our library of inhibitors, we tend to choose very simple readouts that are key to a particular aspect of biology that we're interested in. I have a long-standing interest in T-cell biology, and particularly interested in identifying epigenetic pathways that regulate T-cell inflammatory function. Specifically, IL-17 cytokine expression in Th17 cells, as these are the key players in the pathogenicity of autoimmune diseases such as ankylosing spondyloarthritis. And it was clear from these screens that we observed a number of hits associated with Jumonji histone demethylases, specifically one inhibitor called GSKJ4. GSKJ4 is an inhibitor of the JMJD3 UTX histone demethylases, and these regulate H3K27 trimethylation. So they remove the methylation marks. So JMJD3, therefore, if you inhibit this, what you should expect to see is an increase in H3K27Me3, which is what we're showing here on the ChIP-seq in the graph, the red and the blue. We can see that there's a global increase in the deposition of this mark once you inhibit JMJD3. So the compound is having its effect. We initially started our investigations into understanding the role of GSKJ4 and its role specifically within Th17s using a single-cell mass cytometry CyTOF approach. So CyTOF is a variation of flow cytometry in which antibodies are labeled with heavy metal ion tags, and rather than fluorochromes, you can then read these heavy metals using a time-of-flight mass spectrometry, and we use the Helios system from Fluidigm. We were able to show that 24 hours of culture with GSKJ4 on Th17 cells led to a significant reduction in the numbers of pro-inflammatory cytokines within Th17 cells. Interestingly, we also observed a significant reduction in cell cycle, with the cells essentially stalled in G1 phase. To further investigate some of these findings and identify other functional consequences of inhibiting JMJD3 UTX, we performed bulk transcriptomics. This revealed a significant change in the genes regulating metabolisms within T cells, specifically the induction of a transcriptional program that controls TCA and metabolic pathways. In order to investigate the metabolic functions further, we next undertook detailed metabolomic profiling with Metabolon and GSK and confirmed a significant amino acid starvation response mirroring the transcriptional response we were seeing. We found reductions in several TCA metabolites and observed that mitochondrial function was also reduced, potentially in part explaining the reduction in cytokine production. So epigenetics acts at the single cell level, and we wanted to evaluate the precise effect of GSKJ4 on individual cells within the Th17 cells from patients from ankylosing spondylitis. Despite what we expected to see when we isolated these Th17 cells, we expected to see them as more of a homogeneous population at the baseline. But actually, we observed significant heterogeneity within those cells at DMSO control. When we added the inhibitor to these cells, we observed significant differential gene expression, and we also performed time courses so we could actually study this in more detail. And we found that we actually got a transcriptional program which is very similar to our bulk RNA sequencing. However, we could see a very nice, clear difference in the cell populations, how the cells are reacting between the populations following GSKJ4 inhibition. One thing we could nicely observe was there was a clear separation between our DMSO and J4 treated Th17s, with a clear downregulation in the expression of many genes, again corroborating what we saw at the bulk RNA-seq level. But single-cell RNA-seq actually allowed us to have more granularity over this and specifically look into the individual populations in a lot more detail. When you cluster all of our single cells, you can see that GSKJ4 has a very significant effect on both the highly inflammatory IL-17 and interferon gamma producing cells, in addition to a reduction in markers associated with memory. So we're getting a downregulation of a memory phenotype of Th17 cells. One observation that we identified using our single-cell sequencing data, which we didn't identify during the bulk RNA-seq, was that we observed a clear role for the kinetochore, which supported our previous functional findings that cell cycle was actually significantly affected. And this indicates that GSKJ4 regulates cell cycle in a manner that is kinetochore dependent, and actually we're currently investigating this up in more detail. As single-cell technology has matured and the method has become more routine, I think one of the things that you really also have to think about is that repeats are also very important. I typically see quite a lot of variability within single-cell repeats. Thankfully, in this experiment, we didn't see any significant variability between the different treatments and the different repeats from the different samples. But from analyzing lots of single-cell sequencing data sets, this actually is an issue, and it should really be a consideration whenever you set up any single-cell sequencing experiments. So because we have per sample transcriptome information, one single-cell tool that I actually find really useful for studying biology is the application of trajectory inference or pseudotime. Trajectory inference has it's actually radically enhancing single-cell research by enabling the study of a dynamic change in gene expression in what we would call a pseudotime. So I've used trajectory inference to determine the pattern of a dynamic process experienced by our Th17 cells. In this case, I've modeled pseudotime through as a function of T-cell differentiation. And then the cells are then arranged based on the progression of this process. So the idea is that co-regulated gene expression changes can then be determined and mapped back to the biology associated with culturing GSKJ4. So I use monocle 2 here, and I was able to observe that DMSO and J4 sat at opposite ends of the Th17 differentiation trajectory. Using this trajectory analysis, we see that T-cell receptor signaling is significantly impacted by GSKJ4. And furthermore, we also identified that GSKJ4 may be affecting alternative splicing. And this is actually something that I'm now following up using long-read single-cell nanopore sequencing. which is a method that we're currently in developing, which I won't be going on to talk about today. Another tool that researchers can find useful is RNA velocity, which is a high-dimensional vector that predicts the future state of individual cells on a timescale of hours. So using this tool, we wanted to see if we could predict the effects of GSK4 had on our phenotype and where the cells would migrate to. Unfortunately, the interpretation of this tool in this context is actually quite difficult because we saw large predicted RNA velocities. However, there was a major trajectory flow towards the center of the plot, which focuses on the low IL-17 producing cells. So it wasn't really that useful in this context, but it has been in other contexts that I've used this in. So inhibiting, these are the conclusions for this aspect, inhibiting GMGAD3 UTX with GSKJ4 results in a reduction in inflammatory function in Th17 cells. Single-cell sequencing has allowed us to investigate the dynamic relationship between GSK4 treatment and Th17 subsets. And I think the stress that I want to make is that actually we've learned novel biology as a consequence of using single-cell sequencing that I just wouldn't be able to have understood if I had taken just a bulk RNA-seq approach. So I want to spend the next 5 minutes or so going through other projects that I've been using single-cell sequencing in, and specifically laying out a few lessons that I've learned, which may hopefully be useful for any of you wanting to undertake single-cell sequencing experiments yourself. So one area of research that I work in heavily is in multiple myeloma of bone cancer. We're using single-cell sequencing to evaluate drug responses in bone marrow of drug-resistant multiple myeloma patients. The reason I'm mentioning this is to actually highlight that sample treatment and handling is something that actually really needs to be thought of a lot, because we see actually quite a lot of the time it takes to process cells from these bone marrows from the clinic actually has a major impact on ultimately the sample sequencing quality. However, other types of experiments, you also have to have other considerations. In these experiments, for instance, still in the myeloma field, we've been injecting mice with tumor cells and then tracking their migration within a mouse model. We isolate the bone marrows from these mice and then we process the cells. The consideration for this experiment was how to effectively streamline all of the processes so that all of the mice can be culled and processed in a very time-efficient, dependent manner. Because again, if you don't think about these things before you start the experiment, you're really going to come into quite a significant issues in the long term downstream. So the last example I want to provide is the work that I'm doing in collaboration with the Tendon Seed Network, where we've been isolating cells and nuclei from really, really challenging acellular tissue from tendons. So this is part of the Human Cell Atlas project. On the left, you can see a very rich supraspinatus tendon. It's very cellular rich. However, on the right, you can see that this is a hamstring, which has got a very low cell composition. One of the things that we have realized, is that sample isolation procedures need to be adapted for each individual tissue to yield optimal cell numbers or nuclei numbers. For example, if you don't get this right, you can end up getting a lot of debris within your sample. This is seen here. If you see on the right, the person holding the two tubes, the right tube is very cloudy. This is despite all of the nuclei lysed effectively. And unfortunately, there's just a lot of debris, and this can actually clog microfluidics if you don't remove it. So the way we're taking to get around this is actually to, first of all, modify cell isolation procedures, but also FACS sorting if you're not able to remove this debris. You can see that lots of debris actually has an impact on the ability of the microfluidics to capture your cells. And this is on the left, and you can see you're not getting that many features of RNA and not that many counts. However, with removing this debris, we can actually get very, very good quality RNA-seq data. So the overall conclusions for this aspect is that project planning is really, really important. Get someone who's done a single-cell sequencing experiment on board early. They can give you lots of insight so you don't make mistakes. Again, I see lots of people using single-cell sequencing when it's really not needed. And again, a computational strategy is incredibly important to think about before starting. Get someone who can analyze the data early on board, and they'll also help you develop the strategy for setting up the experiment. And with that, the last thing I want to mention is that suspension cells are really easy. However, tissue is a lot more complicated and involved. With that, I'd like to thank all of those who've been involved, particularly working in my group or within collaborations. And this is particularly Uda Opperman, Martin Philpott, Sarah Snelling, and the rest of the Tendon Seed Network, plus many, many others, as well as the funders for many of the projects. And with that, I'm going to finish and pass that on to, I think, the last slide.
Thank you, Dr. Ayers, Dr. Zajac, and Dr. Kriz for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. Okay, our first question, let me get my questions up. What is the minimum number of cells needed for a typical single cell experiment?
So I can answer this on the probably the statistical side. This is a question that I get asked regularly by many, many people. And it's not an easy question to address a lot of the time because it really needs an understanding of your underlying heterogeneity that you see within the sample, and specifically the smallest cell population that you want to measure. For instance, I do quite a lot of work on cancer, and some of the times you just don't get the numbers of cancer cells required to get enough statistical power. To work out statistical power, there's now quite a lot of very good single cell power calculations that you can do, which you first of all generate pilot data, and then you can feed that data into the power calculations. I think maybe Tom might want to answer this on terms of the hardware.
Mm-hmm. Yeah, absolutely. I'd love to add something on the hardware side there. Of course, experimental design does come first, and I think Adam's done a great job of talking about all that kind of thing. But in terms of absolute cell numbers per sample that can go onto the Nadia machine, which is what I can talk about, our standard single-cell RNA-seq protocol suggests a starting number of cells per sample of 75,000. However, due to the fact that the Nadia is a very versatile machine, we can not only change all of those parameters that I was talking about previously, we can also bring the amount of input from that standard amount of 250 microliters down to around 100 microliters, which means at the same cell concentration, we can go from 75,000 down to around 30,000 cells. So that's what I'd say per sample is the recommended minimum cell input on the Nadia platform. Obviously, you can also go lower per sample if you're using techniques such as cell hashing, as some customers are, in order to load multiple samples into a single cartridge or chip. I think that explains it.
Okay, great. Thank you. Got another question. If you have debris within your sample, what's the best approach to remove that debris? So I can answer that question. The first thing is actually getting your
your protocol optimized to try and minimize the debris in the first place. And this can actually be done by modifying the different types of buffers that you have. Because what we found is you can get away with some amount of debris within that sample as long as the debris is small enough. The problem that we found when we were working with tendon is the tendon tissue is the worst tissue I've ever had to work with in my entire life. It's acellular. It feels like that horrible, gristly bit that you get on steaks that you never want to eat. So you try and you have to really get the methods optimized, because those tissues produce long, stringy bits that basically just block the fluidics. And so the best approach we found, the ideal, is fax sorting. But unfortunately, fax sorting has also its downsides, because you also lose either nuclei or material cells, just because you're not ever going to get 100% efficiency in the fax sorting. We have had experience with removing dead cells using a dead cell removal kit, but the experience, or there's been issues with that in terms of it doesn't really clean everything up 100%.
I think leading on from Adam's point as well on a hardware basis, it's worth me mentioning at this point that generally blockages of the Nadia microfluidic chip are not occurring due to cells. Generally, that can be from overloading of beads or various different things, and that comes from the fact that the Nadia junction itself and the Nadia filter, the smallest point that basically the cells have to flow through, is so the smallest point there is around 50 microns across, which means that unlike maybe some other microfluidic platforms that may have a 30 micron aperture, it is more tolerable to, for example, large cell sizes. So I noticed there, Adam, you were also working, as well as tendons, with some myocardium perhaps, or that kind of cell where you get very, very long multinucleated cells. We do have some users who are also able to run those very large cells on the Nadia platform as well. But obviously, it's all due to exactly how well you get rid of that debris in the first place.
Thank you so much. We've got another question. One of the crucial steps in my experience is having high quality cells. How do you make sure the cells you get from clients are of high quality? Do you provide a kit for isolation to ensure quality standards?
So I could I could maybe respond generally, and then maybe Tom can talk about the Nadia. For me, this is one of the issues that I find constantly is the biggest problem, is actually, and it stems from, I think, getting very good live cells or good nuclei from an experiment. And really the only best approach, the best quality data that we've got, is always tends to come from a fax sorted based method. If it's not single cell suspension from blood, it's very easy. We don't have any issues. The issue that we see a lot of the time is when they're more difficult to extract, or they clump, or for instance, fax sorting or trying to remove that debris is what I can only suggest. Mm-hmm. And also minimizing, sorry, the last thing is minimizing the time it takes to do the experiment is actually really important. Sorry, that's the last.
No, please. So you did touch on an excellent point there, where always consider that if you have a tissue that's difficult to dissociate without turning it to mush and just making all the mRNA from dead cells go everywhere, if that's the difficulty, always consider that there is the possibility of single nucleus RNA sequencing, where granted there are differences between a nuclear transcriptome and the host cellular transcriptome, but if it's the difference between getting a good S-nuc-seq run and a kind of rubbish single cell isolate, you can consider the SNUC-seq in that case, and that is a standard operating protocol on the Nadia as well.
Okay, thank you. We've got another question. Why does Nadia Innovate require Nadia Instrument?
Mm-hmm. So that is due to the fact that the Nadia Innovate is an add-on module that doesn't have pressure pumps in it. So what it does is it hijacks the pressure pumps from the Nadia, and using tubes which are attached to a special breakout cartridge, we then take that pressure and pump it through into a single chip that we can look at using that microscope. Because we only have one microscope, you can only look at one chip at a time, so you might as well just do one at a time. But basically what that is it's taking the internal computer brain from the Nadia and the pumps and using them instead of having to have a whole new system, basically. That's why it's an add-on. And what that means as an extension is that any protocol that can be developed on the Innovate can always be ported back onto the Nadia to be run in full throughput. So whilst you can play around with one sample at a time, once you're done with playing around, that can go back into 8-sample mode, 4-sample mode.
And maybe if I can also add to this for someone who's actually been using, so we got both the Nadia and the Nadia Innovate together. The Nadia Innovate sat on the bench for 6 months, and we thought, well, we've got no reason for this. Why did we get it? Until actually we started to realize the utility of it. We developing our own methods and adding different buffer compositions has made the Innovate method actually a godsend, because it allows us to do a lot more, i.e., we've got these really difficult nuclei in cells. We're changing the cell buffers to actually accommodate those different experiments, and it's actually really been very, very useful for something that sat on the bench for 6 months, knowing what to do with it. We use it probably more than the actual Nadia itself.
That's good to hear. And I mean, if you'd like to read a little bit more about basically a showcase of step-to-step guides of the kind of situation where you might use the Innovate very heavily, you can take a look at our plant protoplast application note on our website, where we've shown how you go from, okay, here's a difficult cell type with a very difficult buffer, very sticky plant buffers, and you need to somehow get that into droplets. It goes in great detail through the process of creating a new pressure profile and of creating a whole new scRNA-seq for your special cell type.
All right, thank you. I have another question. I would be very interested on the experimental design of the J4 DMSO single-cell experiment. How did you overcome batches issues when comparing two conditions?
So the great thing about the Nadia system, compared to the old DropSeq, where you would have to run a sample at a time in series, actually you can analyze I think we tend to run 4 cartridges, but I think the maximum is 8. So maybe, I think it's 8. That allows you to do lots of samples in parallel. So to avoid any batch effects, we always run controls with our treatments, and it's getting the controls correct in an experiment that really gives you the ability to directly compare. Because I've worked on single cell data for probably about quite many years now, and in the old days where you couldn't really do or overcome these batch effects, some of the batch effects were horrific, and there was lots of computational strategies to minimize and get over this. But ultimately, a batch correction is a fudge. It's sometimes a necessary fudge, but if you can run your samples in parallel, that gives you a lot more. They're not confounded, essentially.
Great. Well, that's all the time we have for questions today. I want to remind our audience that those questions we were unable to answer and those that come in during the on-demand period will be answered by our speakers via the email address that you provided at the time of registration. So thanks again, Dr. Ayers, Dr. Zajac, and Dr. Cribbs. Do you have any final comments for our audience?
No, other than it's been a pleasure to work with you, Adam, and we hope that your ongoing efforts to make sort of new and interesting protocols on the Innovate come to fruition.
Yeah, same. It's been.
Okay.
And I just wanted to mention that, well, it's been a pleasure working with all of you, and thank you so much to all the entire audience for taking time to join us at this webinar. And lastly, we hope to see you at our next webinar happening in November, where we will dig more into the data analysis part. So thank you very much.
All right, thank you so much. And before we go, I'd like to thank the audience again for joining us today and for their interesting questions. And again, questions we didn't have time for and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided. We would like to once again thank our speakers for their time today and for their important research. And we'd like to thank LabRoots and our sponsors, Illumina and Dolomite Bio, for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. Please join us in our upcoming webinar, Analyze Your Single-Cell RNA-seq Data from Raw Reads to Insights. That's all for now. Until next time, goodbye.